Okay, so the next talk is by uh, Gianmarco Pinton. He's a professor of biomedical engineering at the University of North Carolina at Chapel Hill. And he studies ultrasound imaging and stimulation, particularly in the brain. And the title of his talk is Ultrasound as a Multi-Hyphenate Ultrafast Super Resolved 3D Neurofunctional.
Thank you. So most of the time when people think of ultrasound, you might think of things like this, so fetal imaging. This has kind of been the poster child for ultrasound for 40, 50 years. It works extremely well. It's cheap, it's portable, it's non-invasive, it's non-ionizing radiation unlike X-ray or CT, and it gives pretty good image quality. So here you can see the profile of the fetus, you can see other features, right? Like the ribs, for example, or the skull, those are bright. Those structures reflect more sound back to the transducer face, so those appear to be brighter than the rest of the body. And it's routinely used. But ultrasound has advanced significantly from these types of images. I mean, look at, for example, the inside of the skull. There's not much inside of here, right? But we know there's a brain in there, and the brain has an incredibly sophisticated structure. And in this particular case, it's completely lost. We don't actually see it with the ultrasound image. So this is an example of something that we're doing today in lab. So using ultrasound images, the same fundamental technology, we can access with exquisite detail the microvasculature of living brains in awake behaving animals, transcranially and non-invasively. So what I'm hoping to do today is to take us through what goes from the regular conventional B-mode image all the way to these types of images and other ways to interact with the brain. The way we make these types of images are with a research scanner. So this is a scanner that's in my lab right now. This is about $1 million. It has 1024 channels, 1024 channels. And what you're really paying for is programmability and flexibility, the ability for a graduate student to sit at this device and be able to use it effectively as a tool. But fundamentally, though, the physics and the transducers that we use are not very different from what you might use from a clinical scanner. So this is maybe $100,000, something you'd find usually in diagnostic imaging routinely in the clinic. The technology scales further down in cost, so you can buy handheld probes. This is a butterfly probe. They're probably on the order around $3,000 or $4,000. Battery operated, connect to your cell phone. Technology, again, is the same. Fundamentally, the physics is the same. So transducers transmit sound into the body, it's reflected back and is generated into an image. That means that you can translate technology very easily from things that might be high cost in a lab to low cost, widely deployable solutions. So what we'll be doing is really focusing, since this is an AI conference, on the machine learning parts that we use to generate these images. The first tool that we're actually going to use is physics. How do we use physics to train machine learning tools to improve our imaging? This resolves one of the fundamental challenges that we have in imaging. When you're imaging the human body, for example, you don't know what the underlying ground truth is. You don't know what's under the skin. That's the thing you're trying to image. So that makes training more difficult because you don't have a reference solution or some sort of way to compare your optimization tools. So our solution to this is to develop physics and physics approaches and simulation tools. So we work both on the physics side and also on the numerical algorithm side. And this is an example of one of the equations that you might develop to represent how ultrasound propagates in the body. I won't go into too much detail, but I will try to highlight some of the essential physical mechanisms that are at play. The first is diffraction. This waves tend to propagate over long distances, about 100 wavelengths inside the body. The other component that's extremely important for ultrasound is attenuation. As the wave propagates inside the body, it gets smaller and smaller, quite rapidly, in fact. And so you might get attenuation that's on the order of even 100 dB. So the sound that's transmitted by the surface of the array is 1,000,000 times louder than what actually is reflected back and recorded. So what that means in terms of your physics and your simulation tools is that they have to have an extremely high dynamic range and an ability to represent very small signals. So essentially the noise floor of your simulation tool must be very low, and the propagation error must also be very low. So this is one of the tools that we've developed. You'll see it a little bit sprinkled through this presentation, but it's something that you can download. What are you going to input into your simulations once you've actually represented the physics? You need to be able to represent the human body. One of the solutions we've found to tackle this problem is through the Visible Human Project, which are cadaveric specimens. These are people that volunteered their body to science, frozen, and then sliced into very thin slices and photographed. And this gives us extremely high resolution, three-dimensional data of the human body, which we can then take and convert from photographs into properties of the acoustical maps. So density, speed of sound, attenuation, nonlinearity. And by doing that, we have a version of the ground truth of what's inside the human body. So this is an example of an ultrasound wave that's propagating. And I may need someone to click on that movie. Oh, there we go. And this is, sometimes I shrink this movie to make the wave more apparent, but I really kept the scales here to scale. So this is about a 400 micron wave, and it's traveling over a distance of 100 millimeters, more or less. So the domain size is a few hundred wavelengths, more or less, in each dimension. That's kind of the size of our problem. The ballistic wave is what you actually use to make an image. That's the thing that actually gets reflected from the targets and is functionally used. But there's a whole richness of propagation that's involved here, right? There's a ton of fluid, a ton of interaction between the wave and the tissue. And even though conventional imaging tends to discard that, it actually carries a lot of information. So we'll be thinking about this from both perspectives. How do we avoid this acoustical noise reverberation? Or actually, how do we use it when it's carrying information? And this illustrates kind of the life cycle of the wave. The wave doesn't always look the same way as it propagates through the human body. As it's bouncing around this near-field tissue, it loses a memory of its past. So this wave field is disorganized after a certain amount of time, right? It's bounced around so many times that it has lost directionality or coherence. And as you get closer and closer to the ballistic component, it regains its structure until finally it has something that's spherical. It's not perfectly spherical. It's a little aberrated, a little bit distorted. That's due to the heterogeneous speed of sound, which is a big source of image degradation inside the body. So once we have an idea of what the human body looks like, once we have an idea of how to model that physics, we can finally generate simulations that propagate these waves through and are actually usable in machine learning applications. So this is an example, a recent example of a data set that we generated. This is for hepatocellular carcinoma. So this is imaging the liver. And so you have to go through the body wall, usually subcostally. And what you can see here is that based on solely the wave propagation physics, we can beamform that, essentially use our simulation tool as an ultrasound scanner, and get highly realistic images of the wave propagation in the body. This gives us then the opportunity to inject this data into a machine learning tool. This is an example of a machine learning tool that one of my students developed, Masashi. And for those of you that do ultrasound, we can go through just a little bit. But essentially, we take IQ data, so in-phase and quadrature data, we split it out into the in-phase and the quadrature part, we condition it with a convolutional neural network, and then we reconstitute IQ data again, and that goes into a differentiable beamformer. So we're re-injecting physics back into our machine learning tool so they learn something about the imaging process. They're not agnostic to imaging. This particular tool knows something about imaging. And then what that allows us to extract is the speed of sound map, which is some version of the underlying structure inside the human body. This is extremely useful for phase aberration correction, which is a way to improve image quality. And so if we compare a conventional high-quality beamformer on the left and then use an iconal beamformer with the speed of sound prediction, we get an improvement in image quality. You can see these vessels, they should be round, but here they're kind of distorted. But here we kind of return them to their roundness. This is very satisfying to a clinician. If a clinician sees this, they're like, okay, the image quality is better, the resolution is better, the contrast is better. I am more confident in my diagnosis of hepatocellular carcinoma. So this kind of approach, what it does, in summary, is to take physics, you train the machine learning tool, and you make a better image that's then interpreted by a radiologist who has higher confidence because the image quality is better. We think we can also do something different and potentially better. What if, instead of trying to make a better image of a conventional B mode image, what if we tried to extract information that's not in a B mode? What if we tried to extract things from the ultrasound physics that's not represented in a conventional image, but that actually is meaningful diagnostically? This is one of the questions that we try to address in lung imaging. In lung imaging, you probably don't think of it as the first thing you might try ultrasound with, right? Your lungs are air-filled organs. It's very hard for sound to get through air. It does not get through air, in fact. So why would you use ultrasound for lung imaging? Well, it turns out that you can have the ultrasound wave carries a lot of information. And in the hands of an expert user, you are able to diagnose lung disease very accurately. So if you spend about 10 years looking at these images, you get better and better and better until after about 10 years, experienced sonographers will be just as good as a CT, which is the gold standard in lung imaging. Unlike a CT, you don't need to take your patient who might be intubated, probably intubated because they have severe lung disease, and you don't need to wheel them out to the CT. You can do this kind of monitoring bedside continuously. Whenever you suspect something might be wrong, you can just get an ultrasound probe on there and do the diagnostic assessment. But it takes 10 years of experience to get there. So if you're selling a $3,000 probe and you like the idea of having ultrasound as a democratized tool, then having a 10-year barrier for training is a big barrier to impose on ultrasound imaging. Okay, so when you look at these images, they don't look anything like the lung, right? The lung is not striped vertically or horizontally, and yet this kind of image carries a lot of information. The lung really looks like this. So these are the alveoli. You can see that there's a very sophisticated microstructure. This is a sponge, essentially, right? That's what your lung is. And these air-filled sacs fill up with fluid as lung disease progresses, and sound can travel into it and reflect multiple times. So these long lines essentially is a misrepresentation of space and time. The echoes inside this pleural surface ring for a long time, but the ultrasound beamforming process interprets that as being long in space. So it's a fundamental misrepresentation of the space. So what we did here is we basically took our abdominal walls and we put the histology from the lung in there. So it's a Frankenstein. We have the abdominal walls from the photographic cryosections, the histology, in this case from cadaveric specimens. But what this allows us to do is to very accurately represent the lung microstructure and the abdominal microstructure together. This is a patient that died from ARDS, so essentially fluid filling in the lungs. The aeration percentage here is around 20%. So only 20% of this lung is air. What we can do in simulation that we can't do in real life is to remove the fluid easily until it's restored exactly at that location to a healthy lung state, which is about 70%. Oh. And when we do that and we propagate simulations into it, we see a very different kind of physical regime that occurs here. On the left, the healthy lung, the sound bounces right off the lung surface. And then if you go all the way to the right, you can see that the sound deep inside the body has propagated far deeper inside the lung. Now, we can clearly see that in these simulations because we have access to information inside the body. But an ultrasound transducer is only at the surface, right? We can only measure information at the surface of the body. And so we don't have access to this when we are using an ultrasound scanner, at least not directly. So we took a bunch of these images and started running these simulations in different cases. And this is an example of an ultrasound B mode made directly from the wave propagation physics that we simulated. And we can compare that to what might be a classical clinical imaging of what's called white lung. So that's one of the terms that a sonographer might use to describe sort of the fact that there's this pleural line right here is dissolved essentially, and you can see the ultrasound sort of streaking through this lung surface, at least on the image. And the exact same behavior appears in our images. So what this means is that the physics that we're modeling in our simulation accurately represents, at least visually, the clinical indicators of disease that a radiologist or a sonographer would use to interpret a lung image. That gave us what we wanted to go to ANIMA and be like, hey, if this looks good to a clinician and it takes them maybe 10 years to actually learn how to interpret these, maybe you can do this too, and maybe you might be able to do it more easily. Okay, so we took a number of cases and simulated. We ran 1.3 simulations for this project, which corresponds to around 10,000 image pairs of ultrasound images. And we injected that into one of the lung ultrasound neural operator networks that Peter Wang developed, Peter in ANIMA's group. I won't spend much time explaining the way this network works because other people in the room know it a lot better than I do. But essentially what we did for this project is we took all the RF data that we simulated it, we put it into a segmentation network that detected the surface into a Fourier neural operator, added some spatial learning, and at the output, instead of outputting an image, we outputted a percentage of aeration. So we predict the image of aeration at the lung surface using ultrasound. And we do this not just qualitatively, but we do this quantitatively. So this is an example of some data. This is the B mode right here. So if you're asked to interpret this and say, well, how much aeration is in this lung? It's a very difficult job to do. A clinician will not be able to actually give you that number. This is our prediction using LUNA, the lung ultrasound neural operators. And then this is the ground truth from our simulation. So pretty close. In fact, on average, our prediction using this approach showed an error of about 4.2%. We're very excited about this. So we ran to our clinician friends and we're like, hey, we want you to interpret these images that are quantitatively express the aeration in the lung and score them as you would a lung ultrasound image. Now, normally, clinically, you score the lung ultrasound on a 4-point scale, 0, 1, 2, 3, or 4, depending on severity. Okay, so it's not a lot of gradations. You get 4 possible gradations from completely healthy to severe. And what we found is that if you actually gave them the images that we just saw, if you gave them the answer on how much aeration was in there, then they actually performed a little bit better than with the B mode. So let me go back. In other words, if you give them this quantitative information, the performance of a clinician is slightly better than if they use that. So that is a good answer, but not a completely satisfying answer. So then what we did is we went back, we got a bunch of porcine lungs. So you can get a postdoc for a year or two and they can get lungs out of a pig, lungs and heart, in fact, so you don't want the lungs to fill up with fluid. And then you fill them up with known amounts of fluid. So you can inject fluid at the lung surface, at the pleural surface. You can flood the bronchi. You can create pneumothorax, so a gap between, an air gap between the two. And then you put a piece of pork belly back onto the lung, an image through the pork belly and into the lung. And this gives you a reference ground truth for the lung aeration. Obviously, you can't do this in people, but we can do this in ex vivo pigs. And that's what finally is able to give us an idea of how good our prediction from ultrasound imaging is using our LUNA approach. And we saw in this case that our errors are extremely low, so 9.4%. So far better than what a clinician is able to interpret, even experienced clinicians. Okay, so I'm gonna maybe just try to spend the next 10 minutes switching topics onto this other part of the work that we do, which is in the brain. And in fact, I'll probably only talk about the imaging and bioeffects part and only really talk about how we support things that inject into machine learning applications. And one of the reasons that we are really focused on the brain is that it has been a rapidly evolving field with ultrasound. I think you saw some of that earlier with Richard's talk, but there's a ton of things that you can do in the brain. You can do functional imaging. You can super resolve things, so improve the resolution by a factor of 10. And those are sort of low power applications, imaging applications. But as you increase the power, you can do different things inside the brain. One of them is neuromodulation, right? So the fact that ultrasound is a mechanical wave has an advantage because it can interact with mechanosensitive ion channels. In other words, you have mechanosensation. You know this because you can feel touch, right? And those ion channels, they are in neurons throughout your body, including your central nervous system. You can increase your power even further. You can open the blood-brain barrier. That means that the tight cell junctions that tend to keep things that you don't want to go into the brain, you can disrupt that. Now, that's normally bad, but if you want to deliver drugs, especially large drugs, that's a very good thing. And you can keep going up in power all the way to thermal ablation. So you can ablate tissue. You can actually destroy tumors, for example, by sending a very high amplitude, high energy beam that's tightly focused to a location, kind of like a magnifying glass that you might use to burn in a sheet of paper, except that you're using ultrasound. So how do we use ultrasound to get a high resolution image? This is one of the techniques that we borrowed from super resolution in optics, and we can do the same thing in ultrasound. So we inject microbubbles into the bloodstream. These microbubbles flow freely into the blood pool. We can localize the center of each of these microbubbles, stack up localizations, and then to get very accurate representations of the microvasculature. So this is about 10 micron resolution volumetrically using the scanner that you saw earlier. This is transcranial and non-invasive in a living animal. And if you add robots to it, I know there's a robotic session coming next, we love the robots, then you can actually get whole brain, at least inside. This is a rat, so you can see the olfactory bulb up here and the cerebellum here. So you're really getting nose to tail on the head, on the brain. One of the applications that we're doing, this is a project with another professor here at Caltech, Mikhail Shapiro, is for brain cancer, both diagnostically and for therapy. So we can image the microvasculature. This is especially interesting for glioblastoma, which tends to be a diffuse tumor. And then we can actually target vessels that feed the tumor while sparing vessels that go outside of the tumor. So the microvascular characterization works in conjunction with focused ultrasound, and those two things are used together to really push the limits of precision in surgery. So that's super resolution imaging up here, which is a technique that's rapidly developing in ultrasound. And we also saw functional Doppler, right? That was in Richard's talk. I won't go over some of those basic things, but the data stream that feeds into these imaging applications is essentially the same. Both these imaging applications rely on high frame rate imaging. So high frame rate volumetric imaging is the thing underpinning these emerging ultrasound techniques. And we can do these two things concurrently and simultaneously because the underlying data can be the same. So this is an example of a functional image with vibrotactile stimulation in a rat. And what we do is we just take a rat and it's like a cell phone vibrator. We just vibrate one of its paws, and then we can use super resolution processing to get a map of the microvasculature, and we can pinpoint the neurovascular unit that's involved in feeling that sensation, right? So that's an M1. And we can do this in real time using single trials, right? So we can see this is transcranial, non-invasive, single trial sort of imaging of the neurofunctional response in that particular motor part of this animal. This has many applications, and one of the ones that we're trying to understand better is how do we actually use this information to read accurately from the brain during behavioral tasks. These are mice that have been trained for reaching tasks, so they'll reach for food pellets. And while they're reaching for food pellets, we can image their brain functionally and get an idea volumetrically of what that reaching looks like. So if you look at the scale down here, reaching occurs at 0. And so activity in the brain starts in the middle as they realize that there's food, and then it moves out to the motor cortex as they start their reaching behavior. So this 3D movie really illustrates the 3D nature of the data, but it is a bit difficult to interpret. So here it is splayed out, but you can see resting and then The mouse realizes there's a pellet. Center of the brain starts realizing this, refractory period, and then motor cortex involved in reaching, and then back to rest. This is single trial data. Our temporal resolution is around 20 milliseconds on those volumes. So we get one functional image every 20 milliseconds. Spatial resolutions, 30 microns. Our data size is about 1 terabyte for 6 minutes. And the size of our data is typically around 100 by 100 by 100, and whatever number of time frames is over here. Okay, so each of these reaching tasks is a single trial which we get. We can compare that to fMRI, where the temporal resolution is usually 1 to 2 seconds. So you really don't have the ability with fMRI to distinguish which parts of the brain are engaged in activities. So we're hoping that by using higher spatial temporal resolution, we can get really down into very specific areas of the brain that are involved in tasks. Why do we want to do that? Well, because ultimately we can also interact with the brain with ultrasound. So that means that we can not just read from the brain, but write to the brain. So we can focus to different areas of the brain where we actually want to interact. This is a SketchUp of one of the devices that we use. So there's an imaging probe right here. Around it, there's a neuromodulation probe. These are 2 different frequencies, a lower frequency for neuromodulation and a higher frequency for matrix imaging. And so then what that means is that we can figure out which part of the brain is involved in reaching, for example, and then target that area with ultrasound, neuromodulate it, change it in some way, and then record the activity and see what actually happens to it. We can do this with high precision, right? We can get down to single vessels in the neurovascular unit, and then we can quantify the amount of the change in blood flow. We can do this in terms of vessel diameter, or also blood velocity, or actually the flow, right? So these are quantitative imaging techniques that get down to very high resolution volumetrically. And when we do that, when we send ultrasound to these regions, we can accurately modify the reaching behavior of these animals. So this is paw tracking. We film these animals as they're reaching so we can track their paws. Excuse me. And we can see that the reference right here is in gray. This is a successful reach. And then when we neuromodulate, we've changed the reaching trajectory, and the reaching trajectory falls short. So we've thrown off the mouse's ability to reach the pellet in many cases, and then this function is slowly restored over time. Okay. And we do this in healthy mice. We can also do this. This is all non-invasive transcranial, so we can do this longitudinally. This is an example from Alzheimer's mice. So we do this also in Alzheimer's disease and look at the progression of Alzheimer's disease and some of the things that we can do in Alzheimer's with neuromodulation. But the fact that this is a non-invasive technique is what allows us to do long-term longitudinal studies, especially with disease progression. Okay, so let me finally connect it to machine learning here. We have high spatial temporal resolution in the whole brain. We can perturb this system with neuromodulation using focused ultrasound at single or multiple locations. We don't only have to send sound at one location, we can do it at multiple locations. This has behavioral outcomes, and we can also combine it with electrophysiology, things like neuropixels. What this means is that we can feed this into foundational models of the brain. We can give this to a machine learning tool that learns what it is about the brain, that makes it function in the way that it does, and what is it about ultrasound that when it perturbs the system, generates an effect. So let me end there with these 3 conclusions. The ultrasound imaging physics is rich and complex. There's a lot of information that's carried by these ultrasound waves, both in terms of the anatomy and the function of the human body. And currently, ultrasound systems capture only a fraction of that information. So if we can develop systems that can capture more of that information, the translational potential is incredible. We can develop these tools to actually go into low-cost ultrasound systems. So we're very interested in combining with machine learning tools and to see what else we can learn from this complicated and rich physics. Thank you.
Very cool stuff. To those of us who are not brain experts, I guess it seems that the neuromodulation is a bit of a blunt instrument in terms of how you stimulate a neuron. And I guess I'm wondering how you translate what you learn by just basically, I assume, vibrating a neuron versus how it might respond to changing external stimuli that it would normally encounter and learn from.
Yeah, so if I were to interpret that question, it is if you have an external stimulus, is that going to be larger or smaller than an ultrasound neuromodulation intervention, for example?
Or have the same characteristics.
Yeah, and that I think is a big challenge for us as a field right now. The short answer is that we do not know. We know we can have dramatic effects. If we use the right sequence, that mouse will stop reaching for the pellet altogether. So that's a pretty dramatic behavioral effect. It's not a small effect. And after that, you can decrease it and change it. You can also get very fine spatial resolution to talk about the bluntness. So the bluntness of this tool is probably around 500 microns cubed. So that's a lot of neurons, but it's also not that many neurons, especially as you scale up to the human brain.
Well, thank you very much. A question. Okay.
So in reference to your neuromodulation results, I saw one of the graphs had kind of like an upshoot in your trajectory after around, I think, was it 12 days? Is it common to see like a refractory response for neuromodulation using focused ultrasound, or?
Is this the graph you're thinking of? Okay, yes. Yeah, so this is the before neuromodulation, and then you're thinking it overshoots, or? Okay, this one. Yeah, so there is a referential thing. So we do this at different times. So this reference kind of shifts a little bit. So in fact, what we end up counting, so that maybe that may be the source. And then what we end up counting is the number of successful reaches from this. Yeah. Thanks.